Okay.
Thank you.
Fabulous. So these are my disclosures. Sorry about that slightly slow start. So I do apologize in advance if you've heard this introduction a million times, but I think probably necessary to just set the scene. The idea is to give you an overview on mutational signatures and to touch on the limitations of this approach. And then I'll talk about some clinical applications at the end. So as a cell turns from being a normal cell into a cancer cell, it doesn't just acquire a handful of driver mutations through its evolution. It acquires a large number of passenger mutations as well that arise because of the various mutagenic processes that are going awry in the cell. So some mutational processes may be perfectly normal, for example, replication errors that arise when cells divide. Some may be due to environmental mutagens that damage DNA of exposed cells, so for example, lung cells and tobacco smoke. Some mutational processes may be endogenous and intermittent, or just the continuous result of defective DNA repair pathways. And each of these mutational processes may leave a characteristic imprint or mutational signature on the genome. When we sequence a human cancer, though, what you see is a final portrait. It's a composite of all the mutational signatures added together, and it may look like a random collection of mutations. However, in the past, it has been shown that if you had genome sequences from multiple tumors of the same tumor type, it is possible to use mathematical methods to extract mutational signatures. So in this case, identify the individual pink rectangles and then to quantify them in each sample. So here it's to determine the size of those arrows. And this was work I did initially at the Sanger Institute. So we presented mutational signatures as substitutions initially because that was simply the easiest class of mutations that we could call using whole genome sequencing in those days. And it's presented in this way. So there are 6 main base substitutions highlighted in the different colors. For each mutation type, for example, C to T here, I've blown it up for better visibility. We can also take the 5' and 3' sequence contexts into account. So there are 4 possibilities, each for 5' and 3', resulting in 16 possible options for each type of substitution. 16 multiplied by 6 gives us 96 possible channels, and a mutational signature is the entire 96 channel pattern, not just the tallest peaks, it's the whole thing. So this is an example of a very common mutational signature defined by overrepresentation of C to T mutations at CpG nucleotides in particular, and is caused by deamination of methylcytosines. It's a perfectly normal mutational signature, and we have it happening in pretty much all our cells. Another example of an endogenous mutagenic process is deamination by the ApoBec enzymes. And this can result in 2 distinct signatures called signatures 2 and 13, characterized by the C to T or C to G mutations happening at a TPC sequence context. Here are 3 examples of very well-known mutational signatures caused by environmental mutagens. Signature 7, characterized by C to T mutations identified in skin cancers exposed to UV light. Signature 4, characterized by C to A or G to T transversions, seen in lung cancers of smokers, and signature 22, defined by T to A mutations in kidney and liver cancers of those people who've been exposed to aristolactic acid. And to give you a sense of what the signatures look like in individual patient samples, we'll look at 4 cancer types: breast, cervix, CLL, chronic lymphocytic leukemia, and lung cancer. So each bar in each of these different tumor types corresponds to signatures that are present in an individual patient. And it's just to give you a spectrum of what it looks like. So signature 1 in green is deamination of methylcytosine, which I mentioned right at the start. It's perfectly normal. So you see it in small amounts in nearly all tumors of all tissue types. It's present in breast, cervix, CLL, and lung. Signatures 2 and 13 are caused by ApoBecs, also present in many samples, but not all of them. But it also shows a diversity in its mutation rate, so ranging from pretty low levels to significantly mutated or hypermutative phenotypes. And you do find some of these tumors with sort of 10, 20, 30,000 mutations almost entirely due to ApoBec. Signature 4, due to tobacco smoke, we find in tumors that have been exposed. So in the cells that have been exposed, so in this case in lung cancers, but not in any of the other tumor types. So a couple of take-home messages here. There's a spectrum. Most tumors have multiple signatures. Some can have hypermutative phenotypes. Some signatures are present in many different tumor types. Some are more specific to a particular tumor type simply because of exposure. So that's just to give you an overview. I will say that the field is pretty new. It's still developing. And so the number of mutational signatures in the literature is certainly not static. It started off as 5 and then 21 and then 30 and now it's 40 plus, and it's going to change again in the next year or so. So this will change as we sequence more cancer genomes in the future. But I will say that I think the big common signatures across many tumor types, I think those have been found. And there are some other signatures, but they tend to be rarer. The second thing to say is that mutational signatures of other classes are coming as well. So there's double substitutions, there's indels and rearrangements. Again, nothing is static here. People are still arguing about what channels should be present in the different signatures. So it's not, it's still a learning process for us all. The third thing to say is that the number of mutational signatures in a data set is subject to some uncertainty. When you're trying to factorize a complex data set, this poses the sort of typical problem of having multiple potential solutions. And the best way to identify the true number of signatures remains a little bit difficult to resolve. But, and regardless of which algorithm you use, and I think there are about 8 different algorithms out there, actually the big main signatures tend to come through regardless of the algorithm that you use. There might be some subtle differences, but the main things, deamination of methylcytosine, ApoBex, smoking, so those big ones, mismatch repair, HR deficiency, they tend to come through. So fourth, the mutational signatures do not all behave in the same manner. Some are subject to more uncertainty than others. And in this PCA plot of attempts to try to extract mutational signatures from a data set, we see that this signature, called signature 17, is easier to extract and forms a very distinctive cluster. By contrast, this collection of different signatures, including signatures 3, 5, 8, 18, and 30, are less discretized and more difficult to separate from one another as distinct signatures. And the arrow points out signature 3, which was first identified in tumors that had BRCA1 and BRCA2 deficiencies, or BRCAness. Signature 3 is frequently used to identify tumors with BRCAness because BRCAness is believed to be an indicator of sensitivity to particular chemotherapeutic agents. But as this plot highlights, it can be difficult to be sure that you do indeed have signature 3 in your data set. Never mind assigning it to individual samples, there's mathematical issues there as well. So stratifying your tumors based on relatively uncertain signatures may result in erroneous tumor classification, especially if you're just going to use the single signature. The substitution signatures, there are 96 elements, remember, not all of them have got very tall spikes, some of them are a little bit flat, so noise can sometimes be classified as a particular signature. So I guess while I'm a big proponent of using mutational signatures, I'd love it to be used. I think we do need to be aware of some of these limitations and be a little bit cautious about making clinical decisions on tumor samples based on just single mutational signatures. We can, however, take all the mutations present in human cancers. So when you do a whole genome sequence, you don't just have subs, you have subs, indels, rearrangements, everything, copy number, and try to help to characterize cancers more accurately by taking the whole picture together. And so in the interest of time, I'm going to try to do this quickly. I'm going to contrast whole genome sequencing data of 2 breast cancer patients for you to see for yourself the distinguishing characteristics of these tumors. It's basically a digital pathology exercise. So this is a breast cancer from a 47-year-old lady. She has over 2,000 substitutions and is dominated by signatures 1 and 5, which are kind of boring signatures. Contrast it to this tumor, which is a tumor from a BRCA1 mutation carrier. And she's got 4,500 substitutions dominated by signatures 3 and 8. And just looking at those wagon wheels or circus plots, they already look very different to each other, right? You're looking at all the whole genome data here. So let's add on indels. The tumor on the right also has sort of different indel patterns with an excess of deletions with microhomology at the indel junctions. This is a classic sign, first described by Maria Jason in MSKCC, associated with BRCA tumors. And then it's also got a large number of a type of rearrangement pattern called tandem duplications. It's also got a large number of copy number losses, which are these pink things going through all the chromosomes, which is loss of heterozygosity throughout the genome. So there are multiple signatures that are involved in BRCA1 and BRCA2 tumors, I think really very distinct to the tumor on the left-hand side. And so we applied machine learning methods to the mutational signatures of cancers from women with germline BRCA1 and BRCA2 defects, and created an algorithm to predict BRCAness called HR-Detect. So a high HR-Detect score indicates a tumor predicted to have a HR defect of BRCAness, and a low score predicts not. And we applied it on these 560 tumors. And what was a little surprising at the time was that we found 124 with high HR-Detect scores. So a high score is a cutoff of 0.7. So 124 of 560 is 22%. We're not talking about 1% or 5% of tumors. It's nearly 1 in 5 tumors. And so we took a closer look. When we looked at the tumors with the high scores, we found the 22 positive controls that we expected, the germline mutation carriers, 22 of them that had inherited mutations BRCA1 and BRCA2. So that was good. Those were our positive controls. But we found an additional 33 women that had BRCA1 and BRCA2 mutation inherited, which they did not know about before this. So either these were from small families, so there wasn't a massive family history, or there was a family history, but that had limited BRCA1, BRCA2 screening as we used to do 15, 20 years ago. And so we would have missed the germline mutations. There were 33 new families of BRCA1, BRCA2 mutation carriers. There were 22 women that had acquired BRCA1 and BRCA2 defects, and 47 women where we just couldn't find a genetic or epigenetic cause of BRCA1, BRCA2. To be fair, we didn't have methylation for all of these women, but we did have it for most of them, and we just couldn't find a genetic or epigenetic driver underpinning the signatures in about a third of the cohort. We decided to then test this algorithm's prognostic performance, because right now it's just an algorithm, right? It's just a mathematical thing. What does it do in terms of predicting clinical outcome? And, you know, for all the genomics work that we've done in the past, most of the time we didn't have treatment or outcome data because it was the best samples in people's freezers. But I had the good fortune of collaborating with Johan Staff and Ockerborg from Lund in Sweden. They have a fantastic setup of very good cancer registries and very good patient collections. And they were collecting all breast cancers in the whole of the south of Sweden. So these are the names of all the local district hospitals in Sweden, where every woman was basically recruited to the ScanB study without exception. So we decided to focus on just triple negative breast cancers because we thought we'd see a lot more of BRCAness in this cohort. And so this is just a high-level summary of the results. First, we found that more than half the patients had HR deficient tumors. I think this is not a surprise. We sort of expected this. It was 58.6% had HR deficient tumors. Second, we found that HR-Detect was able to prognosticate, meaning that patients with tumors that had high HR-Detect scores, so the black line here in the Kaplan-Meier plot, had better outcomes on standard of care treatment for TNBC than those that had low HR-Detect scores. Furthermore, this was irrespective of whether we could find the genetic or epigenetic cause of BRCAness. So we found a genetic or epigenetic cause in about 70%. And the lower plot shows that there is no difference in outcome between patients where we could identify BRCA mutations, and we had methylation and RNA-seq and everything here. So where we could find BRCA mutations genetically or epigenetically, that's in black. It's about 70% of the cohort. Versus those that we could not, which is in gray. So there's no difference really between the gray and the black lines. So taking the mutational signature approach through HR-Detect, we were able to discriminate tumors that have improved clinical responses to TNBC therapy, demonstrating clinical utility of this signature-based tool. This is irrespective of whether you find that driver or not. And here we have really done the full shebang of tests. So a couple of notable additional points from this whole genome sequencing exercise. As Mark has said, I'm a big whole genome sequencing proponent. So in 30% of patients with high HR-Detect scores, we would have missed their HR deficient, their BRCAness, if we performed targeted BRCA1, BRCA2 sequencing alone. So that's that gray line. Second thing is that many current clinical trials stratify patients into different arms based on genetic information, but often this is based on identification of individual driver mutations. Here's a list of driver mutations in this cohort, and I'm just highlighting PIK3CA as an example of such a driver mutation that's often used for stratification. Notice how although it is enriched in the low HR detect cohort, which tend to have poorer outcomes, PIK3CA mutations are not restricted to that category. It's present in nearly all the tumor types. They're seen in the high, intermediate, and the low cohorts as well. So the contribution of information from the rest of the genome, which can affect your a priori likelihood of response, is not taken into account when we do these binary targeted sequencing tests, and could explain some of the more surprising results we see in clinical trials today, right? So people are just taking one mutation, but they're not taking the whole picture into account. And actually, perhaps if they took the whole picture into account, they'll kind of know that there was a higher likelihood of this particular tumor responding anyway to the control arm. The last thing to say is that 4% of this cohort, especially this sort of red group here, the low HR detect scores, and actually in this study, the ones that had a poor outcome, they tended to have mismatch repair deficiency as well. We only found an identifiable genetic or epigenetic driver in one of those patients, but when we did immunohistochemistry, we found concomitant protein losses of either MLH1 PMS2 or MSH2 MSH6 on immunohistochemistry. So there are, so we're pretty sure they had mismatch repair deficiency based on the protein and on the signatures, but we just couldn't find a genetic or epigenetic driver, which is another one of those concerns about just doing a targeted sequencing approach. So that's just to give you a sort of an overview of sort of the use of whole genome sequencing and mutational signatures in our population-based cohort, where we developed an algorithm and then tested it in the cohort. I'd like to end on a case study to demonstrate why it's important to interpret the whole cancer genome and not to just look at one or two things, just the targeted sequences, not just individual mutations. So I do have the permission from this patient to talk about him. His name is Joshua Barnfather, and he's 30 years old. He was diagnosed with a rare inherited genetic condition called xeroderma pigmentosum type C. So he's the kind of patient I see in clinic because I'm a clinical geneticist and not an oncologist. So this is an autosomal recessive condition. He's inherited 2 abnormal alleles from both parents. And historically, these kids with abnormal XP genes would get a lot of UV damage, and they'll succumb to their disease because of the UV damage. They get skin cancers and they die. So the XP genes are involved in the nucleotide excision repair pathway and is absolutely necessary to fix UV-related damage. I was serving as part of sort of the DNA Repair XP service in London. These kids get a fantastic service. They're seen from birth. They get fantastic education and photoprotection. So they now live to adulthood, like Josh. Unfortunately, in January 2017, he presented with this violaceous lesion on the medial aspect of his left eyebrow, and it was diagnosed as an angiosarcoma, which is a rare tumor of the blood vessel lining. He had a wide local excision, no radiotherapy, and by September he had lymph node mets and was commenced on typical sort of chemotherapy for an angiosarcoma, but he didn't respond and was entered into the TAPAS trial, an RCT of pazopanib, a multi-targeted receptor tyrosine kinase inhibitor, blocking tumor growth and inhibiting angiogenesis. But he also did not respond to this. And by the end of June, he had infiltrative sort of base of skull mets with direct cranial extension. By July 2018, really, this man was dying. And I was asked to assess the whole genome sequences of his angiosarcoma. And so here's a circus plot, and you note instantly that there's a very heavy burden of mutagenesis throughout this genome. These red dots are C to T mutations. He's got a really boring set of drivers, a CDKN2A and P53, and doesn't have the sort of typical drivers associated with angiosarcoma of PTPRB, PLCG1, or MYC amplification. But his mutation profile and signatures are rather interesting. So he's got these 800,000 mutations, mainly C to Ts. This looks, to all intents and purposes, if you just pass it through me, I would say this looks like a malignant melanoma. Because malignant melanomas tend to have hundreds and hundreds of thousands of C to T mutations that look like signature 7 due to UV light. But remember, he is an XP mutant and he's unable to fix UV damage. So this is likely to be the appearance in all his normal cells. He does, however, have this mutational signature, which is signature 10, associated with mutations in polymerase epsilon. And so we did have to do a little hunt because what he had was we did find the driver, but it was really, really subclonal, so it was dismissed by most of the algorithms. But we did find an S459F mutation in POL-E in a subclone. And actually, if you look at the blue and the pink lines, they're actually much smaller than the red lines because the POL-E signature is present in the subclone as well. So this is interesting because POL-E signatures, like mismatch repair deficiency signatures, they're associated with response to checkpoint inhibitors. We did have to seek corroborative evidence to permit the doctors to give him the treatment. So we did PD-L1 staining, but we did it in the primary tumor, and this came back as negative initially. And don't forget, if this is a subclone, this may just be a false negative. So we just missed it. So we then did it in the Mets, and indeed this showed 60% positivity. So this is the UK. To get access to the drug, this poor man had to crowdfund his own treatment. He's the bravest man I know. He smiled throughout. This was him towards the end. It was really quite hard to look at him with his lymph node met sort of deforming his face. I'm pleased to say pre-treatment, you can see multiple lesions throughout his body and his liver, encasing his sort of mediastinum as well. And after 3 cycles of pembrolizumab, there was significant reduction in the volume of disease and partial metabolic response. By about July 2019, pretty much everything had melted away, and he left hospital and was back at work until COVID-19 broke out. So that was a very nice story, anecdotal, but really just trying to explain why it's important to interpret the whole patient, not just the specific mutation, but also the germline as well as the somatic genomes. So that was my presentation. It was an update on mutational signatures, the concepts and caveats, and then clinical applications, both in a population-based study as well as in an individual patient. There's a couple of last slides. I would have liked to talk about this, but in the interest of time, I don't have time, but just to say that we are doing some systematic experimental studies to validate these mutational signatures. We already have a paper out on all the environmental mutagens, and then we've got a paper in bioRxiv on systematic knockouts. We also have a website that you're very welcome to kind of explore. All the signatures we generate, we dump out there, including all the knockout ones, they're all there already. And you can even perform mutational signature analysis on our website. So I'd like to end by acknowledging the people with the enormous foresight to have started the whole genome sequencing exercise in the noughties when it was incredibly expensive, and all the funders that supported them, because I would not have the career I have today if they did not plan this exercise. So it's all really down to those people who had the foresight. And this is my team who have the time of our lives playing with the data that's being generated, not just by ICGC, but by heart rate people like you guys. So yeah, keep producing the data and putting it out there. Thank you very much. Happy to take any questions.
Thanks so much, Serena. That was an amazing tour de force. I think you're a couple of minutes under. So if I might take the prerogative to ask a question that you hinted at the end, the XP patient accumulating those mutations from their germline. I think that's what's changed remarkably over the last few years, is understanding somatic mutagenesis in healthy normal cells. We've had this knee-jerk reaction to sequencing tumors, finding signal and saying, ah, it's tumor. But what are your thoughts on where the field's at now, where it needs to go to?
Right. So that has been really exciting in the last few years. Sequencing of normal tissues has shown up that there are loads of drivers in our normal tissues. There's loads of little clones as well. And there are, unusual distributions of some of those drivers. Notch1, for example, in the esophagus, P53. And so it's the interpretation of the things that we find, right? When you see a driver, actually, are we certain that was a driver, or was that just something that we've seen because it's just very frequent? P53 mutations are just extremely frequent. In that tumor, is